Today I'm going to tell you a little bit about our project to identify or to profile the RNA isoforms in human brain that are associated with risk in neuropsychiatric disorders. So we know after several years of genome-wide association studies that these have identified hundreds of risk loci for the neuropsych disorders. However, most of these are in the non-coding regions of the genome, and we think that these are acting on disease risk through altered RNA expression and through alternative RNA splicing, which was explained a bit earlier by Bernardo. But that basically what is going on here is that there are different coding exons being joined together in our mRNA that are potentially leading to different protein products that have a different function, or maybe leading to RNA decay. So previous research in our lab in the CACNA1C risk gene for schizophrenia has identified hundreds of isoforms that were previously unknown. So we've decided to go ahead and sequence a further 31 risk genes, which you can see in the bottom left-hand side of the screen there, for schizophrenia, bipolar disorder, major depressive disorder, and autism spectrum disorder. And what that Venn diagram is showing is just the degree of overlap for some of those genes and their risk for those disorders. So we took healthy post-mortem brain tissue from humans across 7 different brain regions from 5 individuals, giving us 35 samples all up. The brain regions that we did cover were primarily frontal cortex regions, as well as cerebellum and caudate nucleus. We extracted our RNA. Hopefully that comes up. Extracted RNA, did our cDNA synthesis, and what we did then was long-read amplicon sequencing using primers that were designed in the 5' and 3' UTRs to capture as many of the basically the reference isoforms as possible. We did this using the LSK 109-110 kits with the barcoding 96 kit, and sequenced and subsampled our reads to a minimum depth of about 8,000 reads per sample. We used our in-house bioinformatics pipeline DiscoAnt, which again is using Bamboo, which was mentioned earlier. And for our visualization, we used Isomix, which has now been changed to Isoviz, which I'll try to remember to say that. So these are both tools that are going to be available very soon. So some results covering all of the risk genes that we have profiled so far. Our amplicon size ranged from about 1.1 kb for the bipolar risk gene LMAN2L, all the way up to just under 11 kb for the schizophrenia risk gene CSMD1. In total, we identified 929 isoforms. 775 of these were classified as either novel in catalogue or novel not in catalogue. So that is where novel in catalogue is where it's using known junctions, whereas novel not in catalogue has at least one novel splice site. And what we're looking at here is basically all of our genes together and the proportion of reads on the x-axis there that are assigned to either the reference or the novel isoforms that we have found, just really showing basically the amount of unknown isoform expression that we are missing for some of these genes. So novel splice sites that I mentioned, we found 113 novel splice sites. The majority of these are using the canonical splice donor and acceptor GTAG. So a quick couple of examples of those. For the depression risk gene, DCC, there's a couple of transcripts that we identified, shown by the black tracks there, with a GT donor, a novel GT donor that added an extra 35 nucleotides, which contained a premature termination codon in it, probably leading to nonsense-mediated decay for those isoforms. Again, a novel splice acceptor AG, showing for the schizophrenia risk gene CPT1C, adding another 98 nucleotides, also probably leading to NMD. And you can also see another track in there that has a retained intron. So we also identified several novel exons within our data. We found about 21 of those. Again, most of those were containing a premature termination codon. Some of those were frame retaining. Some of those were found in the 5' and 3' UTR regions, and a couple of them contained a premature termination codon that was less than 50 nucleotides from the final junction, so we don't think those are necessarily getting marked for NMD. So next we wanted to have a look at a further breakdown of our proportion of reads assigned to each of the novel isoform subcategories as identified through using Scanty. Basically anything here that is in grey was a reference match, And then anything else in color was either assigned as a novel splice site, a retained intron, or the other two categories I mentioned earlier. And what this is really showing is that the nanopore amplicon sequencing can find all these different kinds of isoform subcategories. So a great example of just an individual gene here now, not all of them together, is the schizophrenia risk gene ATG13. And what we're looking at here is actually an isovis representation of this gene, the known and novel isoforms that we've found for it. On the left-hand side, you can see our transcript representations with the box and the line through. And on the right-hand side is all 35 of our samples with a heat map basically off the count data out of our sequencing. And what this is really nicely showing is that using this technique, we can find relatively highly expressed novel isoforms, in this case for transcript 18, which is skipping exon 12. And you can also see as well in the other box a little bit of variability in the 5' UTR for transcripts 1 and 3. Another gene I want to briefly touch on is the neuronal growth regulator 1. This is a cell adhesion molecule that's involved in neuronal outgrowth and connectivity. It is a shared risk gene across schizophrenia, autism, depression, and obesity. Basically, we amplified this gene using 2 forward primers to capture 2 different start sites with a single reverse. So it was all together in one reaction. The expected sizes for this was around 2.7 kb, covering 7 coding exons. Again, we have another isovis representation of our findings here. We detected 5 novel isoforms and 2 novel exons for this gene. These 2 novel exons were between exons 6 and 7, so towards the end of the coding sequence here. The first one in transcript 1 was 42 nucleotides in length and actually retained the open reading frame, so it's still using the canonical start and stop. The second one here was 58 nucleotides in length and had a premature termination codon in it, but less than 50 nucleotides from the final junction. So together, these 2 transcripts made up about 20% of our assigned reads for this gene, which is not insignificant compared to the canonical, which is shown by the ENST tag there. So just to make sure our novel exons are real, we designed primers flanking those 2 novel exons, did some Sanger and PCR, so you can see in the orange and blue boxes there, confirmation in our gels that the exons were actually there. And as an added bonus, our Sanger for transcript number 1 lined up really nicely. You can see our Sanger sequencing on the top row there, our long reads in the middle, and then high vertebrate conservation, just giving us a little bit of a backup that exon is real. So we have a couple of novel isoforms, a couple of novel exons, and we kind of want to have a little bit further look at what they may be doing. So we just decided to do some protein structure prediction using AlphaFold. So here we're looking at the canonical isoform. It's 354 amino acids in length. You can see on the left-hand side there, N-terminal running through 3 immunoglobulin-like domains through to the C-terminal. AlphaFold itself, if you're not familiar, gives you it's colored by its prediction confidence. So in blue is high confidence, and then anything in that sort of orangey yellow color is a low confidence, so it can move around a little bit each time you would predict it. Highlighted in that red residue there is actually a GPI anchor, which would normally anchor that protein into the membrane. So NEGR1 was a recent study by No et al., I think it was, that showed that NEGR1 is interacting with the leukemia inhibitory factor receptor, which in turn is involved with the control of expression of the cytokine lipocalin-2 involved in hippocampal neurogenesis. And the NEGR1 knockout in mice actually show depressive and anxiety-like behaviors when compared to the wild type. So we went ahead and did our prediction for our first transcript. So if you remember, that is the one that had an additional 14 amino acids in it that is retaining open reading frame. And you can see there in the pink residues on the right-hand side that those would be basically included now. That is a sort of lower confidence estimate there. But you can see that it shifts the position of the GPI anchor, which I can't say for sure, but obviously this may impact how the NEGR1 is interacting with that particular receptor. The second one here, transcript number 2, which contained the premature termination codon, basically removed that GPI anchor entirely, potentially removing that interaction. So there's those 3 overlaid on top of each other. I don't know what your idea of fun is, but mine is to turn this into a little movie that spins around. And in conclusion, long-read amplicon sequencing can identify known and novel full-length isoforms in human brain. Novel exons that we do find have a high chance of being validated, which is just really great. We identified 775 novel isoforms in healthy control human brain across our 31 neuropsych disorder risk genes. So what's next for us is to continue our validation of the remaining novel exons that we have. I want to take a look at, see if we can do isoform switching using antisense oligonucleotides. But really what this work is doing is laying the foundation to improve our understanding of how these RNA isoforms are impacting protein function and therefore neuropsychiatric disorder risk. So I would just like to thank Nanopore for inviting me here to speak, and the Clark Lab and our funders. So thank you.